Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Streamline Targeted LNP Development to Improve Preclinical Translation. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank PhosphorX, who developed the content for this presentation. PhosphorX is the go-to partner for helping innovators advance complex therapeutics through innovative drug delivery platforms, including lipid nanoparticles, LNPs, polymeric nanoparticles, PNPs, and polymeric microspheres. With scientific creativity, deep technical expertise, and a highly collaborative scientific team, they help innovators engineer target-appropriate biodistribution and scalable, reproducible delivery systems. Now, I would like to introduce our speakers for today's event. Our moderator for today's discussion is Dr. Alan Horhoda. He is an industry veteran in nucleic acid therapeutics and nanoparticle delivery with nearly 20 years of experience advancing RNA medicines and delivery technologies. Also joining us today is Dr. Nicholas Boylan, Senior Director, Scientific Services at PhosphorX, where he leads a team of scientists and engineers specializing in the development and optimization of novel drug delivery systems. Dr. Syed Reza, Licensing and External Innovation Alliance Management at NOF Corporation, brings 20 years of experience in biopharma and specializes in nanomedicine technology. And Dr. Michelle Belrose, a study director at Neosome Life Sciences, where she leads preclinical study design and execution across a broad range of therapeutic areas. But now, without further ado, I'd like to go ahead and hand the presentation over to Dr. Alan Horhoda to begin. And you may begin when you're ready.
Thank you for the introduction. The last few years have ushered in a resurgence in targeted LNP development, catalyzed by clinical demonstrations in the immune field, with progress made in other tissues and cell types as well. We often joke that all LNPs are targeted. It just depends whether a chemist or a patient's body added the ligand. But making, characterizing, and testing actively targeted LNPs brings additional challenges not faced in passive approaches. We have a great group of speakers here today who will help us walk through some of those differentiating factors and how they help clients navigate targeted LNP development in a time, cost, and resource efficient manner. So with that, maybe we kick things off with you, Syed. At the core of all of this is an LNP amenable to targeting in the first place. We know it's not as simple as adding a ligand to a generic LNP and hoping it works. So how do we need to think about those base LNP properties to set our ligands and ultimately the payloads up for success?
Yeah, that's a good question. Obviously, what the ligand is doing is helping the LNP arrive at the cell surface. And then possibly by that interaction, if the receptor undergoes endocytosis, also allowing the LNP to be internalized. But after that process, the composition of the lipid nanoparticle is really important to allow the RNA to be released and to be expressed. And then you also have to consider the safety aspect, the tolerability of the lipid nanoparticle. So essentially, I think the first priority obviously is to choose the lipid components that are known to be well tolerated. And this can come from lipids that have been used in other clinical settings or with NHP data on the lipid nanoparticle, just using passive lipid nanoparticle compositions. But once you have a lipid that meets those criteria, then you can formulate into composition. Then the other question is the composition tuned and optimized for that particular cell type to allow the endosomal escape and release to occur? And here we find that by changing the lipid composition, the other helper lipid, or even the identity of the ionizable lipid plays a role in helping that lipid nanoparticle fuse with the cell membrane and also to allow endosomal escape to occur. So just by optimizing the lipid composition, sometimes we can get a 5x to 10x improvement in RNA translation. And so one of the ways we can approach this is you can take the target cell type. Let's say if we're looking at T cells, you can look at primary T cells in vitro and try to just screen the lipid composition alone to identify the optimal one for entry into targeted T cells, into T cells, sorry. And then we can add the ligand on that and then reassess if that works in vivo. So that is essentially taking a very complex problem and breaking it down into its individual parts for a solution.
Thanks. Are you seeing bulk particle properties start to shift as you think about extrahepatic delivery in terms of things like size or surface charge that make some base LNPs more amenable to targeting than others?
Yeah, I think size is an important parameter, but currently a lot of the effort is on delivery to immune cells. And where immune cells reside in the spleen or in the lymph node, for example, they are the blood vessel pore side, the fenestrations are pretty large. And so even if you have a lipid nanoparticle of, let's say, around 200 nanometers, which is commonly seen when monoclonal antibodies are added to the surface of a nanoparticle, those are able to penetrate and achieve delivery. But if we are then looking at applications where we need to deliver LNPs to the lung or to other cell types where the endothelial passages are much smaller, then we still need to retain and limit the nanoparticle size. So the way to go about doing that is to use antibody fragments, which don't increase the particle size that much, or with peptides, aptamers, or even small molecules. And so those are other strategies that are available to maintain small particle size for those applications.
You'd mentioned composition a bit earlier as you think about ratios or chemical choices. Can you speak to some of the properties or the key components that really seem to drive the differentiation from a traditional passively targeted LNP to something that's geared a little bit more towards that active targeting approach?
So there are certain things that we know already. One is that for active targeting, we're often using, most likely you're going to use some kind of reactive chemistry on an intact lipid nanoparticle. So the first concern is ionizable lipid. Is the chemistry compatible what we're using? So for example, for our disulfide containing ionizable lipids, they're not terribly compatible with melamine chemistry. So we use other click chemistry approaches. We also are looking at non-covalent interactions to do active targeting. So I think that's one of the primary concerns, just to make sure that you have chemical stability. And then we have some frontier research that's indicating that the fluidity of the lipid nanoparticle composition also plays a role in maintaining stability of the ligand on the surface. So we're just starting to kind of scratch the surface to understand how for example, the melting point, the transition melting point of a nanoparticle affects its ability to retain the ligand on the surface as well. But I think in the future, such know-how will be very useful to making more effective LNPs.
As you think about the different ionizable lipid choices, some of the challenges that predictability has faced, are we getting better as an industry in understanding, which chemical choices are going to set some of these active targeted formulations up for success versus others?
No, not really. I think that's one of the big problems, that there isn't a way to rationally navigate the different chemistries. It's really very much a trial and error approach. And I think that's where a lot of the risk and cost comes in. But I think working with either, if you have people in your organization that have some experience on a lipid chemistry, lipid technology, that can really help. Or if you work with external partners such as Phosphorex or with us at NOF, then we have a lot of know-how on working with these technologies, so we can help to navigate some of these challenges.
What are some of the other trade-offs that you might face in terms of, balancing liver detargeting versus, say, performance in extrahepatic tissues? Are there challenges in that balancing act that you're seeing?
Yeah, I think the trade-off is quite significant because the lipid nanoparticle does have a natural affinity based on a size and characteristics to be taken up to the liver. So even if we have some formulations that can reduce, for example, ApoE binding, and so they will allow more delivery to the spleen. And then with active targeting, we can increase the level of precision to get to specific cell types in the spleen as well. But there is a trade-off in overall activity of the RNA expression compared to, just a naked LNP that'll go right to the liver. I think that with some of the newer lipids that we have, we've been able to sort of engineer a property that essentially detargets hepatic delivery, and those are showing some promise. I think that we would really want to establish further safety data on those lipids, but I certainly think that that's another area that we can get to. Again, the problem with lipid technology is a lot of it is trial and error, so you can't rationally design a lipid to detarget the liver. But certainly by doing a lot of screening, we're able to achieve certain benefits in that area.
Thanks, Ed. And maybe that's a good place to switch over to Nick. And so, Nick, as we assume we're getting stronger and stronger base LNPs, the next question really is, what do we do with them from a targeting perspective? There's ligand choice, conjugation, analytics, but maybe just from a high level, why is targeted LNPs worth it? What are those advantages that we're hoping to gain as we think about an actively targeted versus a passively targeted LNP?
Absolutely. Let me just first pull up a slide as a visual guide here. So as we discussed earlier, the non-targeted or passive LNPs, as depicted in the left panel, rely on the formation of the protein corona. This is formed by the absorption of serum proteins onto the surface of the LNPs, with the classic example being ApoE-mediated uptake by hepatocytes via the LDL receptor. As Syed highlighted, to achieve passive targeting to other organs such as the lung and spleen, the lipid composition can be modified to alter the LNP surface chemistry, which directly impacts the types of serum proteins found in the protein corona. Corresponding changes in the protein corona are responsible for altering the in vivo biodistribution of the passively targeted LNPs. Therefore, for extrahepatic targeting, it's generally worthwhile to perform some optimization on the base LNP formulation to take advantage of any endogenous passive targeting before implementing active targeting strategies. However, a potential downside for passive approaches includes demographic-dependent variability in the protein corona. In addition, some patient populations may lack the target receptor, as is the case for patients with certain liver diseases who are deficient in the LDL receptor. For hepatic delivery, these deficiencies have been successfully overcome by targeting the LNPs by incorporation of GalNAc ligands onto the surface of the LNP. This effectively redirects the LNP uptake via ASGPR, which is a receptor found in high density on hepatocytes. Some additional benefits of targeted LNPs include greater cell and tissue specificity, which may lead to reduced off-target exposure. There's also potential for improved intracellular delivery, which can lead to higher therapeutic efficiency and potential for dose reduction. An element here from a biological standpoint is ensuring that the target receptor is internalized upon binding and not recycled back to the cell surface. TLNPs also open access to difficult to reach cell populations such as immune cells, and there's huge potential in the personalized therapy space. But of course, all these benefits come with some downsides, such as the added formulation and CMC complexity.
Yeah, you'd mentioned GalNAc, and it's sort of been the gold standard ligand for the last 10 or 15 years in the targeted space. It's not simple, but it doesn't have some of the complexities that other ligand classes have. Can you touch on some of those different ligand classes, whether it's antibodies, peptides, sugars, and which ones are showing some of the best promise as you think about those next generation of ligands?
That's a great question. As each ligand class has distinct strengths and limitations, at phosphorics, we believe the best choice depends on a variety of factors, including the target disease indication and practical issues such as manufacturability and immunogenicity. Antibodies are great due to their high binding affinity, clinical use, and general availability against a wide range of targets. Therefore, they can be a very useful tool when exploring the biology and for applications where the target receptor is expressed at low densities. However, the relatively large size can be a limiting factor when conjugating the surface of LNPs, as well as their manufacturing complexity and associated costs. Next, you have your STFEs and Fab formats, which are about 3 to 6 times smaller than full antibodies, while retaining the variable binding domains. Due to their smaller size, there's potential for less impact on LNP size, which can lead to better tissue penetration. They're also more amenable to screening applications. However, in some cases, they may be more susceptible to aggregation. Currently, a lot of our work is shifted towards the use of the nanobodies. which are about 1/10 the size of conventional antibodies. In addition to their smaller size, nanobodies, due to the lack of glycosylation, can be produced in a cost-efficient manner by microorganisms. We can also purchase nanobodies from our manufacturer with a C-terminal cysteine for site-specific conjugation. This ensures that the ligand has proper orientation on the LNP surface. As we've already touched upon earlier, the use of sugars or carbohydrates as targeting ligands, such as GalNAc, are another promising approach. However, other examples include mannose for targeting dendritic cells and macrophages. Some advantages of carbohydrate-based ligands include their excellent safety profile. However, downsides include their limited application to cells that naturally express the target receptor, and there's potential for endogenous sugar molecules to compete with the LNPs for receptor binding. In addition, individual monovalent binding affinities are relatively weak, and therefore a multivalent display on the surface of the LNP is important to increase the overall binding strength through multiple ligand-receptor interactions, which is referred to as the avidity effect. Finally, peptides have great potential as well due to their smaller size and ease of manufacture via synthetic routes. However, some potential downsides include lower binding affinities and susceptibility to enzymatic degradation, but these can be overcome through chemical modification to improve their stability, as well as multivalent display on LNPs to result in stronger binding affinities through the avidity effect.
Yeah, you'd mentioned avidity a few times there. And, with nanoparticles, it's not that clean sort of one ligand, one payload relationship that you see with other therapies. How do you guys approach that ligand density question and sort of optimize for maximum engagement for a given target on a cell of interest?
This is somewhat class dependent, as ligands with lower binding affinities, such as carbohydrates, peptides, will need to be displayed at higher densities in the LNP in order to benefit from the multivalent avidity effects, whereas ligands, including nanobodies and mAbs, can be incorporated at lower levels. One important consideration is the orientation of the ligand on the LNP surface, as ligands displayed in a random orientation will have some of their binding domains facing away from the LNP, while others might be facing towards the LNP and or be sterically hindered. For small ligands manufactured via synthetic routes, it's relatively easy to incorporate the conjugation chemistry handles at sites that do not interfere with binding efficiency, while also ensuring proper orientation. In addition, site-specific conjugation can also be achieved for larger protein-based ligands, but it's typically not a standard option for your off-the-shelf monoclonal, and therefore represents an important consideration when selecting the ligand class. Having said that, Phosphorix's approach is to identify the range of ligand densities which can be displayed on the LNP without negatively impacting quality attributes such as size, PDI, cargo encapsulation efficiency, as well as other factors including product stability and manufacturability. Once we have identified the design space, tLNP test articles can be prepared with the suitable reporter cargo system, such as mRNA encoding luciferase or mCherry. These LNPs can have low, medium, or high ligand content, which can then go into in vitro and in vivo studies at Neosome to assess target cell engagement, expression efficiency, and biodistribution, determine the optimal ligand content on the LNP. And then just to give a quick snapshot or preview of some internal data that we're generating here at Phosphorix, this slide just highlights a actively targeted LNP that's been fabricated with low, medium, high VHH content. And here we purposely selected this range just to demonstrate the impact that the addition of this VHH or nanobody on the surface of the LNP can have on particle size. So as you can see here, going from the plain or naked LNP, which started off about 95 nanometers, kind of incrementally increasing the amount of VHH in the formulation. We see the size progressively increase, right, from 100, about 140 up to 200 nanometers. And again, this just highlights that, more is not always better. So we recently, manufactured these test articles, fully characterized them, and we have sent them over to Neosome to initiate the in vitro and in vivo work. And so what we're anticipating here is likely the lower medium formulation here in terms of VHH content is probably going to provide the best therapeutic efficiency. And likewise, the particles prepared with the high VHH content, this presents challenges from a 0.2 micron filtration standpoint. So there's certainly some manufacturing considerations there. But again, just to show kind of a sort of snapshot of some data we're working on, just illustrating the importance of one, identifying, what range of ligands can be incorporated. And again, this is very class dependent, but then also having the in vitro and in vivo data to iterate and fine tune those formulations.
You mentioned shipping in there. As you think about the stability of these particles, they're more complicated than sort of passive formulation. So, how do you think about the stability of that formulation, both from a vial stability, but also, post-injection stability, to make sure that that ligand is still, on the particle displayed correctly and can function the way it's designed?
That's a great question. And ignoring chemical stability for the moment, the stability of the ligand surface presentation relies primarily on the nature of the PEG utilized for conjugation. In the case of PEG lipids, the PEG shedding effect in which the PEG lipid dissociates from the LNP in vivo is well established and depends on the length of the fatty acid tails. Therefore, ligands conjugated to, say, DMG PEG, which utilize fatty acid tails of 14 carbons in length, will shed more quickly than ligands conjugated to, say, DSG PEG, which has a fatty acid tails of 18 carbons in length. From a chemical stability standpoint, buffer conditions such as pH, osmotic strength, and in storage conditions, time, temperature, they're acceptable for the free ligand, would be good reference points for ensuring stability of the ligand post-conjugation. However, these are just guidelines, as the storage conditions for the LNP must also maintain other critical quality attributes such as size, PDI, as well as cargo encapsulation efficiency and integrity of the RNA cargo. In terms of quantitation, Phosphorix routinely develops analytical methods for quantifying both the conjugated and free ligand content. However, we utilize a phase-appropriate approach, and therefore, for early development purposes, these methods typically aren't stability indicating, and therefore we truly rely on the in vitro and vivo readouts at Neosome to confirm that the conjugated LNPs maintain their targeting ability.
What are some of the other common things that trip people up? What else should be on our radar as we think about developing targeted LNPs?
Lots to consider here, and I'm sure we'll get into some of these items in a little bit deeper context during the question and answer section. But just to kind of highlight a few, first, it's really critical to understand the biology, and then this will inform the ligand class, which then informs the conjugation strategy. And just to highlight there's typically two routes. So we can prefabricate LNPs, so referred to as post-conjugation, where the LNPs have the chemical handle on the surface that enables the subsequent conjugation to the ligands, whether this is thiol-maleimide-based chemistry or click approaches. Alternatively, we could prefabricate the base LNP and then separately conjugate the ligand to, say, PEG lipid in micellar format, and then through ligand insertion, incorporate those ligand conjugated PEG lipids back into the lipid nanoparticle. And there's kind of pros and cons with both of those approaches. From the formulation side, not only do we need to identify the base formulation in terms of ionizable lipid, helper lipid PEG content, as Syed highlighted, but again, it's determine what is the chemistry and how are we going to incorporate that ligand into the formulation ultimately. And then finally, there's the how much ligand do we want to incorporate into the formulation, and how does that impact the quality attributes for the LNP itself in terms of size, polydispersity, cargo encapsulation efficiency. But then from manufacturing standpoint, as well as analytical, we need to confirm that there's no free ligand present in those final test articles. This can be controlled both through the stoichiometry of the chemistry being utilized, and then also purification approaches such as size solution chromatography. And then with those final test articles, we need to have some confidence in terms of their storage stability, the ability to ship these test articles to external partners such as Neosome. And this all really lands heavily on the analytical team, again, in terms of the standard analytics for LNPs, but then also those more specialized methods for quantifying the ligand content. We are routinely running various methods there that are orthogonal in nature, both plate-based methods, but also liquid chromatography-based methods, a lot of size of solution chromatography. And then recently we've invested in the CloudSpec instrument, which enables us to do, I'm going to call it simplified UV-Vis-based measurements, but typically when you have a particle present, that's going to scatter light and interfere with those measurements. And so this CloudSpec technology, through their scatter-free absorption measurement, allows us to collect, say, UV-Vis to quantify both protein and cargo in the intact LNP without effectively disassembling those particles.
Thanks, Nick. You talked a lot about some of the analytical characterization, but as you think about it, that's often just the start of validating performance. So maybe this is a good time to bring Michelle in, because we ask these particles to perform in a very complex biological milieu, and the physical characterizations aren't always a great predictor of ultimate performance. So maybe you can help us understand how various in vitro models and in vivo models can help clients de-risk the outcome, especially as you think about species to species factors and some of the complexities that targeted ligands bring that maybe you don't see in a passive formulation all the time.
Thanks, Alan. Yes, in vitro and in vivo models are very useful for when you start to evaluate the performance of that LNP. And I first want to start with in vitro models, as this is often where we start when we receive LNPs for evaluation. These tools are useful for evaluating cell targeting and biological function before moving into your preclinical model. Each in vitro model has its distinct strengths and limitations, and using multiple complementary approaches can help de-risk translatability. For example, cell-free assays are useful in evaluating specific molecular interactions. For example, your targeting ligand interacting with your target receptor. And cell-based assays are useful for assessing LNP binding, uptake, and payload delivery. Primary cell lines are a particularly useful tool in this context, as they often provide greater biological relevance and are generally more predictive of in vivo biology than immortalized cell lines. Just to elaborate on primary cell-based assays more, one major advantage of this approach is the ability to evaluate the performance across a variety of cell types and species, which you touched upon. For example, comparing LNP binding, uptake, and delivery in primary cells from multiple species can help identify species-specific differences in receptor engagement and cellular uptake delivery. And these data help guide preclinical model selection and also provide important context when interpreting your in vivo results. Additionally, in vitro assays are valuable for distinguishing receptor-mediated targeting from nonspecific uptake. This is what Nick touched upon before. When we receive those LNPs from phosphorics, we can then look at that targeting ability. And this can be done using cells with varying levels of endogenous receptor expression or engineered cells that express or lack the receptor of interest, allowing changes in LNP binding and uptake to be attributed to the targeting ligand on your LNP.
Yeah, you mentioned uptake and uptake specificity. For a lot of LNPs and modern payloads, just getting to the cells isn't enough, right? You really need to have that intracellular uptake and endocytosis, intracellular trafficking. Can you walk through how you help clients sort of evaluate those various aspects that ultimately have to be successful for your drug product to work?
That is a great point. As you said, receptor engagement and cell association don't always translate into functional delivery. And here, those cell-based assays are also a great tool for the uptake and productive delivery. And measuring this does depend on the question you're trying to answer. Commonly, at the start, fluorescent labeled nanoparticles with quantification by bulk fluorescent measurements, flow cytometry, or microscopy are commonly used because these provide a streamlined way of seeing your LNP taken up and being internalized. And choosing this appropriate readout depends on the stage of development and the specific question being addressed. For example, bulk fluorescent measurement provides a rapid, high throughput assessment of LNP association and can be particularly useful for screening studies or evaluating association kinetics over time. Flow cytometry provides a more quantitative and single cell assessment across large populations and can be combined with phenotypic markers to evaluate how uptake varies with cell type or receptor expression. And microscopy provides a more spatial approach where you can evaluate intracellular localization and distinguish internalization from surface association and characterize uptake patterns. Beyond uptake, intracellular trafficking can be assessed using approaches such as galectin recruitment assays for endosomal escape, while functional delivery can be measured through that reporter gene expression or other cargo-specific functional readouts, such as protein expression, genome editing, et cetera.
Thanks. I'm not sure there's any, easy answer, but as you think about, different endpoints and predictability, right, are there some that are proving to be, more predictive for, functional nucleic acid delivery than others?
The most informative endpoint is going to assess functional delivery that's dependent on the therapeutic modality you're testing and the mechanism of action of that payload. These generally include readouts that measure the cargo expression or the downstream biological activity rather than uptake alone. So the reporter systems I discussed about are a really streamlined readout for that delivery efficiency because it depends on the functional activity of the reporter to be expressed. But when you're talking about untagged cargo, functional delivery really depends on what cargo you're using. So you can look at cargo expression by ELISAs, flow cytometry, other protein-based assays. When you're focusing on gene silencing or gene editing applications, your functional endpoints are tailored to the mechanism of action, such as target knockdown, editing efficiency, or downstream pathway modulation. And these can be assessed with your molecular approaches, such as quantitative PCR or sequencing-based methods.
Yeah, we mentioned a lot of in vitro characterization, but obviously in vivo is probably the more important data set to gather, and it's a lot more complicated system too. You've got biodistribution, pharmacokinetics, and other things that won't show up in cell-based assays. So, how do you think about that transition from in vitro assays to in vivo assays, and what are the factors that people need to keep in mind as they're thinking about these targeted LNP testing?
Yeah, as development progresses into those in vivo studies, your goals typically shift from understanding the behavior in isolated cells, as you mentioned, to determining whether that LNP distributes in the complex biological system. And this can really be assessed by biodistribution and PK studies that rely on complementary imaging and bioanalytical approaches. And this can help you determine whether it reaches the intended tissue and also the intended cell population. I'll elaborate on these, starting with imaging approaches, which can include fluorescence and bioluminescence imaging. Fluorescence enables visualization of the labeled LNP biodistribution in live animals and excised organs. And bioluminescence provides an additional functional readout because it relies on reporter expression for the bioluminescence and therefore indicates intracellular delivery. Additionally, flow cytometry can further characterize biodistribution at the cellular level by identifying which cell populations have associated with or internalized the LNP, and this provides insight beyond the whole organ distribution. The bioanalytical methods, including mass spectrometry and other molecular assays, quantify LNP lipid components or nucleic acid cargo in blood and tissue samples. And this provides quantitative PK and tissue distribution data that complement those image findings. When you're designing your biodistribution preclinical study, we often encourage people to use serial blood sampling and longitudinal in vivo imaging because this allows the biodistribution and PK profiles to be evaluated within the same animal and reduce inter-animal variability while also providing insights into circulation, tissue accumulation, and clearance over time.
Yeah, it's a challenge for LNP delivery is just that localization is not functional delivery. So, how do you think about parsing those out in terms of differentiating simple tissue accumulation from truly functional delivery?
That's an important question. An LNP may accumulate within an organ but fail to achieve productive intracellular delivery for multiple reasons, for example, due to uptake of off-target cells, endosomal entrapment, or degradation of the cargo. And in many cases, the same assays we use in vitro that I discussed a few minutes ago are also used on the tissues that are collected from the animals. The additional value of the in vivo setting is the ability to connect those tissue level readouts to the pharmacodynamic responses across the whole organism. And this provides further evidence that the delivered cargo is biologically active. So, if we're thinking about this, depending on the therapeutic mechanism, this may include changes on protein expression, cytokine production, pathway activation, immune cell responses to the LNP delivery, or restoration or suppression of your target gene function. And together, these functional endpoints provide stronger evidence of successful delivery than tissue accumulation alone.
As we push past the liver for extrahepatic targeting, what are some of the preclinical models that seem to be popping up as being more predictive than maybe some of the legacy models that we had 5, 10, 15 years ago?
There isn't always going to be a single preclinical model that is universally most predictive. And we like to encourage that the best model depends on the biological question being asked. And we have clients think about the important considerations, including target tissue, therapeutic mechanism, and whether the targeting ligand and receptor interactions are conserved across species. Conventional mouse models do remain highly valuable for early formulation optimization, biodistribution, PK studies, and preliminary safety assessments when that target biology is conserved between species. When the target ligand or receptor is human specific, humanized mouse models become increasingly important, and a lot of progress has been made there. One example of using humanized mouse models is NSG mice engrafted with human PBMCs. And these are commonly used to evaluate human immune cell targeting, receptor specificity, and in vivo delivery to human immune cell populations. Similarly, mice expressing humanized versions of the target receptor or using transgenic mice can be valuable when species differences would otherwise limit interpretation of preclinical data. And going further, it's important to consider the disease model when you're evaluating efficacy when you get to that point. One great example of evaluating efficacy is in oncology. Humanized mouse models and other disease-relevant tumor models are useful for evaluating targeted delivery to human immune cells, in vivo CAR generation, and looking at that anti-tumor activity.
Thanks, Michelle. I know there's probably countless topics that we haven't touched on as we think about targeted LNP delivery. So maybe this is a good time to open things up to some questions and let our panelists dive into.
Yes, well, thank you so much for this very insightful presentation today. At this point, as we move into the Q&A session, I do want to direct the audience's attention towards the handouts module, where you'll find some additional documentation related to today's presentation to download for some further reading. Also want to encourage you to keep sending your questions or comments for the Q&A portion of the webinar. If you're attending via GoToWebinar, please use the questions window. And for those watching on LinkedIn Live events, we'd love to hear your thoughts into the comments section. Now, before, though, we take a look at some of the many audience questions we've gotten so far, we would first like to take a moment and hear a little bit from you, our audience. Appearing on your screen right now is a polling question. You can participate in this by selecting either option and then clicking submit. If you're attending via GoToWebinar. For those on LinkedIn Live events, still, we'd love to see your comments coming into the comment section. The question that we're asking you, our audience, right now is, are you currently developing targeted lipid nanoparticles, TLNPs? It's just simply a yes or no question. We'll give you some time to answer that. And while you're doing so, we'll go ahead and take a look at some of the questions we've seen come in from the audience. Now, the very first question that I have for you, our speakers here, someone wants to know, they do say that they are new to the field, but can different routes of administration, maybe SC, help to detarget liver tropism and uptake?
Thanks, Ryan. Maybe I'll get us started there. Yeah, definitely, a great question. I think certainly with sub-Q, we're going to have better delivery to that local site, so it's going to reduce the Cmax exposure, systemically, and certainly limit the amount of LNPs that go to the liver. However, it really depends on the end goal. So if your end goal is to target tissues that require access via the systemic route, of course, going sub-Q is going to have some limitations there. And just give some examples, right? If you're trying to target maybe the lung or CNS systemically, then going sub-Q, of course, may have some limitations there in terms of biodistribution. But I think at a high level, it is important to consider, right, all those various routes of administration when considering, your biodistribution and how that impacts the target tissue and cell type of interest.
Perfect. Thank you so much for your answer, though. Now, let's take a look now here at our audience's answer to the question again of, are you currently developing targeted lipid nanoparticles? It looks like we have 45% of our audience saying yes, and then 55% coming in for no. This is a very interesting result we have here.
Yeah, I agree. And I think and maybe we've kind of sort of selected, right, for individuals that are actively developing targeted LNPs. So of course, I think the percentage is like a little bit higher than I would have expected, just based on I guess incoming inquiries, right? A lot of our work currently is with non-targeted passive approaches, but we are seeing growing interest, of course, in the targeted field, and this is just going to continue growing, I think, in the future. So, yeah, an interesting polling result there.
Perfect. Thank you. Let's go ahead and pull up our next poll for the audience. So this next poll, in this case for our audience, you can actually select multiple answers and then click submit, more than one that you feel apply to you. The question we're asking is, what targets are you pursuing? Answer options to choose from are muscle, lung, immune cells, or other. As we have this up, let's take a look at another audience question here. The question I have here wants to know what immunogenicity concerns are associated with ligand-conjugated LNPs?
Well, I think I can take that. So I think the biggest concern is really just the type of ligand that you're using, that, if it's an antibody, it should be humanized. But I think for many of the current applications, that's not a huge concern, because you're often able to take antibodies that are repurposed from therapeutic or diagnostic indications and then repurpose them for targeting. And in general, the ability to create antibodies that are, fully humanized, non-immunogenic, well tolerated is quite good out there. I think the one unknown right now is what happens when you have antibodies attached to a lipid, because now you're essentially creating a new type of epitope. And one thing I'm just worried about is that if you have very high density of protein molecules on the that is immobilized on the surface, that can present a way to cross-link weakly reactive anti-drug antibodies, and thereby initiate a complement cascade. So I think that's something that we have to watch out for, particularly with LNPs that have either high PEG content or high antibody or protein on the surface, where I think that might be something that could pop up. So definitely something to keep an eye out.
Perfect. Thank you very much. Let's take a look at the results for this next poll from the audience. We have here again answering what targets are you pursuing. We have 21% people choosing for muscle, 29% for lung, 63% then for immune cells, and 46% of people selecting other here.
That's probably not a huge surprise given the interest in immuno-oncology, autoimmunity these days. The number of CAR papers and posters you're seeing out there, and hopefully some good clinical data soon, I think speaks to the interest in that area.
Perfect. Yeah, thank you. Now, let's pull up our next poll for the audience here. This next question is wondering what types of ligand classes are you working with? This is another one where you can select as many as apply to you and then click submit to register your answer. Again, that's for our GoToWebinar audience, and for those on LinkedIn Live events, we see your comments coming in, so thank you for that. Keep doing that. The answer options for this are MAB, a CFV or FAB, VHH, sugar, or peptide. We'll leave this up for a bit and take a look at, the next question from our audience for you here. So next question is asking, during the manufacture of TLNP, just how does ligand insertion compare with post-conjugation approaches?
Yeah, maybe I'll get started there. And comparing these two approaches, right? So post-conjugation, well, effectively what we'll do is prefabricate the LNP, and then by incorporating a PEG lipid with the appropriate chemical handle, those LNPs will have, say, a maleimide on the surface. And so this approach is, we routinely use this at Phosphorus. It's highly amendable to screening applications where we can kind of fine tune the amount of maleimide on the surface of the particle. And then with the given batch of particles, we can screen various ligand types and really tailor amount of the ligand that's incorporated on that formulation. Maybe tying back into Syed's comment on the immunogenicity, important aspect here that's applicable for both post-conjugation and ligand insertion is the capping of any unreacted maleimide groups, just as an example, because if these are retained on the formulation as they go in vivo, serum proteins such as albumin could potentially conjugate to the surface and result in complement activation, right, clearance of the particles, and potentially unwanted immunological side effects. But having said that, ligand insertion, alternatively, here we're separating that conjugation chemistry so that that's occurring away from our sensitive RNA cargos, just as an example. So in that workflow, we would prefabricate the base LNPs, but there's no need to incorporate, say, the maleimide chemical handle on those base particles. Separately, we can essentially take our, let's say, DSPE-PEG maleimide, prepare, under aqueous conditions, it's essentially a micellular format of those pegylated lipids, and onto the surface of those micelles, we can conjugate our ligands. So this gives the opportunity to one, perform that conjugation in the absence of what tends to be very expensive cargo. We can also sterile filter that if there are any aggregates formed, and then do a quick QC check before performing the subsequent step that's really critical, and it's the insertion process, wherein these ligand conjugated PEG lipids are incorporated back into that base LNP. And so this process is very time and temperature dependent. So we need to be at a temperature that's above the melting transition point of sort of the composition of the LNP from a lipid standpoint, so that these hydrophobic tails on the PEG lipid can incorporate into the surface. So again, two kind of separate workflows, some pros and cons for each of these. I think at the end of the day, the analytics remains critical in terms of quantifying, one, is there any remaining free unconjugated ligand present? In the case of ligand insertion, we also have to verify, one, is there any remaining micelles present in that final drug product, and effectively, one, purify away those micelles. And then either approach can have some ramifications in terms of workflows and stability of your drug product, and trying to maintain the best conditions, right, for your cargo, and likewise your ligands that you're using. And maybe just a closing thought here as well. Occasionally, this is more for peptide or small molecules. In some cases, we might need to use an organic solvent to effectively solubilize those and facilitate the conjugation reaction. So in those types of applications, the ligand insertion method has its advantages, because addition of organic solvent to that conjugation reaction would effectively destroy the LNP if it were present during that step. So again, I think there's really a lot to consider there, but just kind of touching on some of the differences.
Perfect. Thank you very much. Let's look at the results here from our poll. Again, of what types of ligand classes are you working with? We have 38% of our audience choosing for MAB, 33% for SCFV or FAB, 48% VHH, 19% sugar, and 48% peptide. So thank you to our audience for participating in that. We have one more poll for everyone here, and it's popping up right now. This question we're asking, and it's only choose one answer and then click submit for this, is what's the biggest challenge that you face in tLNP development? Answer options are ligand selection, conjugation scale-up or CMC, analytical characterization, or preclinical testing. And while we have our final poll up, let's go to the next audience question here. Someone wants to know, how does the galectin recruitment assay inform intracellular delivery?
Yeah, I can elaborate on that. I mentioned galectin recruitment assays when I was talking about in vitro models, and this helps as a useful readout for intracellular delivery because it assesses the endosomal membrane disruption, which is one of the barriers for successful nucleic acid delivery. So galectins are cytosolic proteins, and they can bind glycans that are exposed upon endosomal membrane damage. So if you use a reporter cell line that expresses fluorescently tagged galectins, such as Gal3, Gal8, or Gal9, and pairing that with fluorescent microscopy, you can visualize the galectin localization, and you'll see a shift from diffuse cytoplasmic signal to distinct fluorescent puncta when that endosomal membrane is disrupted. And this just gives a confirmation that you're getting intracellular delivery versus just binding. And then you can go downstream and look at your functional nucleic acid delivery.
Perfect. Thank you so much for answering that for our audience. Now let's look at our audience question result here for the what is the biggest challenge you face in TLNP development. We have 19% of our audience choosing for ligand selection, 44% then for conjugation or scale up or CMC, 31% for analytical characterization, and 6% choosing preclinical testing. Again, thanks to our audience for that. I don't know if anyone has any thoughts on those results.
I think, fundamentally scale up CMC does become a problem because the know-how to create, to prepare functionalized nanoparticles at the bench scale is quite widespread. Many innovators, researchers are able to do it. The problem really becomes in creating something that's much more highly engineered and controllable and scaling that up. And I think that's where a lot of us are struggling with. And I think some of those problems, if they can be solved, it'll really accelerate the rate at which early academic bench scale discoveries can be translated into usable products.
Yeah, I think Nick mentioned a couple of the insertion strategies. There are some conjugation approaches that work great for sort of papers and posters. There's other strategies that are a little bit more amenable to the things that end up having to make their way towards patients. So if you haven't kind of thought through that transition from early proof of concept, what gets you the data that's interesting therapeutically, but also going to be meaningful from a development perspective, you can end up in some corners that you may not have wanted to paint yourself into.
Yeah, I think one of the technologies I'm keeping an eye on is, so Nick talked about, and there was a question about surface conjugation versus post-insertion. But one of the areas that I think is quite promising and exciting is the use of non-covalent linkage strategies to create functionalized nanoparticles. So an example is, for example, using streptavidin biotin to take a protein molecule and attach it to a immobilized surface which has biotin on it. We commonly use that for affinity chromatography. Of course, streptavidin is not safe. It's highly immunogenic. You can't use it in a therapeutic setting, but there are variants of that approach that are being evaluated. So I know, for example, there are companies that are using bifunctional antibodies where they'll have a small molecule type of domain in the lipid nanoparticle that's included when the LNP is being formulated, and then they can simply incubate it with a bifunctional antibody so that one end binds to a small molecule and the other end is your targeting moiety. There are also reports of double conjugated nanobodies, so a VHH that's conjugated to make a dimer at a C terminus, with one having affinity for the bait or sort of the anchor in the nanoparticle, and the other one is towards the ligand. So there are these kinds of innovative approaches that sort of combine chemistry with molecular biology techniques and really leverage the know-how that's 20, 50 years old in terms of creating these kinds of pull downs and try to apply them to LNP. So I think those could be very exciting because that might present us with a sort of a kit-based approach where you can just make your LNP, independently vary the formulation, and then incubate it with your ligand and get it to work. So I think some kind of a breakthrough really is needed to have a better way of making targeted LNPs. That's just my opinion.
Mm-hmm. All right, thank you very much for sharing your thoughts and opinion on that one. Next question here wants to know, do you find an impact of particle size on the efficiency or response depending on the animal model you may be using for preclinical testing?
Yes, so I think it's well known, maybe just using hepatic delivery as the example there, there can be differences in terms of, what is the optimal particle size between species. And again, with hepatic delivery, in mice, generally particles 70, 80 nanometers, work well for uptake. Whereas as you transition to non-human primates, smaller is a little bit better just due to the size of the penetrations and the requirement for smaller particles to be able to access those tissues. So that certainly consideration. So I think it really just highlights the importance of one, understanding biology and how that might change as you across different species and work towards, human subjects, of course. So I think we're just continue to learn and more in that space. But yeah, go ahead, Al.
And I think a lot of that historical information has just been so heavily focused on the liver as you think about the last 20 years of LNP or nanoparticle delivery. So it'll be interesting to see how those rules get written for the spleen, the kidney, some of these other tissues that are now in the forefront of where people are trying to take these payloads.
All right, very good. Thank you so much for sharing. Again, now this next question here, it starts by saying that you showed some data indicating that increasing Fab ligand density can promote aggregation in LNPs. The question is, does increasing density also promote corona formation in vivo? And if so, does this corona end up hindering targeted delivery?
Yeah, absolutely. Another good question here. Yeah, I would almost treat these as two separate things in the sense that, as we're incorporating more and more ligand on the surface, there can be a crowding effect, right? Where, maybe it actually reduces the efficiency of binding to the target receptor. Or of course, if the particle size is increasing, through aggregation, these sorts of mechanisms, then I think that immediately raises some red flags about, the stability of the formulation. And likewise, when it's administered in vivo, is it going to be able to circulate long enough, right, to reach the target, tissue and cell types? So generally, we want to kind of steer away from that. Maintain a small particle size is generally better, assuming we have sufficient, ligand density. And then, the second element there in regards to the protein corona, I think it does sort of circle back to the base composition and the formulation, right? What ionizable lipids are present, what helper lipids, what is effectively the, maybe the size as well, but also like the surface chemistry of the particle, and that's directly going to impact the types of serum proteins that attach. However, I'm certain that the ligand on the surface of the particles is also going to impact that. I think by our standard workflows is always to conjugate on the end of a peg chain to make sure that these ligands are a little bit off the surface of the particle and therefore don't get buried under the protein corona. And maybe one element there could be, length of the peg, right? So that could be sort of tuned to determine how far off the surface that ligand is. But I think, all these factors need to be, considered, right? With the base formulation, what is its inherent tissue tropism, and likewise, when we attach that ligand, ideally we're not putting too much that we're causing stability issues with that formulation. And then ultimately, we need to test and confirm, right, that we have the appropriate receptor engagement to facilitate, right, that selective uptake in the target cell and have that productive RNA delivery. A lot of factors to balance there.
Mm-hmm. All right, very good. Thank you very much. Next up, someone's just wanting to know, how does the length of the PEG linker affect the efficacy of conjugated antibodies for LNP targeting?
Well, I think that's what, Nick, you were talking about just now about using PEG linker. I think, so the length of the PEG linker, if you're performing surface conjugation, generally using a 2000 molecular weight PEG, and we can get good conjugation take place with that. So we don't see a need to really change the PEG type there. But one of the things you want to think about is that if you have a high PEG content of non-activated, non-conjugated PEG on the lipid nanoparticle, that PEG tends to drive the lipid nanoparticle to condense upon itself. So it's a little bit of a smaller particle size is driven by increasing the PEG content, but that also tends to disfavor the addition of a large macromolecule on the surface of that lipid nanoparticle. So it's almost sort of like a squeezing effect where the high PEG content is trying to squeeze the nanoparticle down, and it can pop that protein off. So if you're using a large monoclonal, that can be an issue. But one of the ways that we would recommend assessing that is simply by performing incubation of your LNP with with serum, and then do like a 24-hour incubation and analyze the components after that. And that gives a pretty good idea of the stability of the nanoparticle in blood and those dynamics of lipoprotein absorption and protein corona formation on the LNP. Okay.
Excellent. Thank you so much for that answer, and all of you for all of your answers today. But we have reached the end of our time for the webinar. No to our audience, if we couldn't attend to your questions, the team at Phosphorex will follow up with you. Or if you have some further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen if you click the exit button, and your participation is appreciated, as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing that recording and more. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.